Okay, good morning. We're ready. Looks like it's just you two. I don't see anybody else online, but maybe somebody will sign up later. Remember what I said in class on Mondays, that Wednesdays are going to be online, so we're going to be doing this kind of a little different. This is the online setting that you would see. Is it hearing? Yes, good. All right. I'm always concerned that I'm staying too far back from the microphone. And I told you guys the other day, when I'm on campus on Wednesdays and Thursdays, even though this is an online class, I can, y'all show up. I'm more than happy to stand in the classroom and do it. If not, then, you can do it remotely. Now, next Wednesday, I won't be here. So class next Wednesday will be remote. When you sign in, you'll see me, and I'll be at my house. I won't be here on the following Wednesday. I'm sure I'll mumble too much with the same tweet, far away from the microphone. So, first thing I would ask anybody, if y'all have any questions, anything about Blackboard, anything. I was going to say, anybody have any problems getting on Collaborate? If you guys aren't on, nobody else did. So, it seems like that was pretty easy. Any problems we have with class, downloading the book, the PDF? Any problem with that at all? Let me know if you have issues with that so that I can make those kinds of corrections. Now, it's going to look like I'm going to be trying to talk to the whole class, right? This is going to be recorded, so it's going to be a little weird, which is very less in the room, but I think we can probably make it work. All right, so I put this back up again to make sure that you have a chance to practice through those different types of reasoning. They are easy to confuse, and probably like when we did the metric conversions the other day, the best way to do them is just to do bunches of them. And I always tell students, and you heard me say it already, the self-assessment part. You go online, and that's the beauty of having that, and you type in examples of types of reasoning or examples of types of inductive and deductive reasoning, and you will get pages where people have gone through and give you examples. So the more of those you practice, the better you'll get. Remember, the big thing about the difference between them was one makes a prediction, one does not, right? So one makes a very giant statement, right? And then the other one makes a prediction out of that. So that's kind of maybe where you start from the frame to think about whether it's inductive or deductive reasoning, okay? So do practice those. Not only just those, but also ones that you might find online as well. And let me know if you have any that come up and you just don't know. Say it says it's inductive and I don't understand why, because they're not always going to be worded exactly this way, right? I kind of worded them in a way that, kind of helps you guys get used to the way inductive and deductive reasoning works, but you won't always find them worded that way. So it might be a little bit confusing. Now, because my PowerPoint didn't load this morning for some reason, I'm having to use the PDF, so this is a little bit cumbersome, but you guys, it'll be fine. We'll make it through here. So I want to go down to here. This is where I wanted to go. This is familiar maybe to some of you. You're saying, well, I can't see it yet, so you're going to have to show it to me. There we go. Is that readable for y'all? Okay, good deal. All right, so if you're looking at your first assignment in class, the scientific method assignment, you probably recognize this. This is the lead into the question, or at least the essay part of the question. That scientific method assignment has a series of multiple choice and some ordering kinds of questions. But at the very end, there's an essay. And I'm expecting an essay. I'm expecting a written answer, all right? So I don't need a book. But I did want to go over it with you because in many cases, sometimes this is the first time you all have encountered this whole idea of breaking down a scientific experiment, figuring out what part is what. Where's the observation? Where's the hypothesis? So we'll start with this, and this is in the later part of the 19th century. A disease known as beriberi began happening to people in the Dutch East Indies, and beriberi causes things like body weakness, lethargy, inhabitants. They don't eat, they become very tired and fatigued, and they can occur, fatalities can occur, usually often from heart failure, right? Which makes sense. If you're not burning any energy, nobody will pump the heart. And it's always kind of a funny thing with biologist medicine when people say, what was the cause of death? We always say, the heart stopped. That's always the cause of death. No matter what. No matter what happened, you fall off a building, your heart stopped. So it's an interesting thing to say that, if you get to the point where something that causes your heart to stop, you pretty much know what's going to go on. So originally investigators thought that this thing was a bacteria, right, causing the disease, this beriberi disease, and they set up an experiment where they injected some chickens. So you have to think about it here. What we're doing is using the chickens as a placeholder for human beings, obviously. It's not horribly ethical to go inject human beings with disease, right? So we're going to inject these chickens, and what we're going to do is we're going to take the blood from the symptomatic individuals. Now, You got to make sure that you dissect this out and don't get lost, because when I get a lot of these answers from students, they start talking to me about being injected with the blood and all that. It's not the blood, folks. It's the bacterium that's in the blood. Remember, the original hypothesis here originally, investigators felt it was a bacterium causing the disease. That's the hypothesis. I said the other day in class that often hypotheses have a question mark, right? Or often interrogative. Now, this one does not have a question mark, but you can see quite clearly that's what they're doing. Investigators thought, felt, questioned, came up with an idea, guessed scientifically with what was causing the bacterium, right? So we're going to take the blood out of symptomatic human beings and we're going to inject it into the chickens. Because come on now, if there's a bacterium in the blood and it causes this disease among humans, I know maybe what you're thinking is because it causes disease among chickens. Yeah, chickens get the same disease too. There's a lot of diseases that pass back and forth. You get rabies from dogs. All right, so if we inject these chickens with the blood and it has the bacterium that's causing very people that are sick, then the chickens should be what? If we inject it with blood with the very bacterium, what should happen to the chickens? Of course, right? If he has a disease and it's some blood-borne disease, a bacterium, and I inject it into you, well, guess what? You're going to get the disease as well, right? That's the transmission that's going to occur. So that's a good test to see if it really was a bacterium, right? Injected into the chickens. If the chickens get sick, then obviously it must have been a bacterium. If the chickens don't get sick, then it's something else. Let's take a look and see how we can make sure, right, that control part that I talked about. Because we got to make sure that we're controlling for all the variables here, right? So the chickens were injected with a symptomatic individual. So the injected chickens became sick, okay? But what they did smartly here is they had two groups of chickens. That second group of chickens is the control group of chickens. Remember Monday, back in my days, we had an experimental group. So those are the chickens we're going to inject, all right? And then we have a control group. And what we're going to do with those chickens is we're not going to do anything to them. We're just going to leave them alone, all right? So we had these injected chickens, and they became sick. So did the other chickens that hadn't received the injection. So here's my control group. Here's my chickens. Remember, the difference in a control group is one variable. One variable. One variable is different between those two. Two groups of identical chickens, right? And two groups of chickens that are living in the same habitat and eating the same thing. One difference between them. One. Just one. Some of them are getting the injection of the blood that maybe has bacteria, if that's what it is, and some were not. So we have a problem here. Why did the chickens who didn't get injected with the blood, presumably having the bacteria, get sick? Well, maybe it was a bacterium that was airborne and was passed from one chicken to the other. We didn't test for that, so we can't really answer that question. We know that it's in the blood, so it's a blood-borne disease, but we didn't really witness any chickens exchanging body fluid in this place. So it probably wasn't something like that. So something else is going on. So our initial hypothesis was the bacterium. What are we going to do now that we've pretty much, or at least estimate that it wasn't the bacterium? What are we going to do to our hypothesis? Yeah, we're going to try something else. We'll come up with another. Remember, that's a series of things we go through the scientific method. Right, now here's some of the terms I was talking about earlier you may or may not be familiar with, and they're important in understanding the way science works, especially when we present data graphically. I asked you, what are the independent and the dependent variables? Now, those of you who have had statistics, I'm sorry, I'm going to draw in here so folks online can see this too. Those of you who have had statistics have probably seen those terms before. You may remember them, you may not remember them. And when it comes to looking at data graphically, it's really important to understand which one is which. So let me go ahead and do that. Let me draw a graph, or the axis to a graph. There we go. And along the bottom of the graph, what we're going to do is we're going to put the variable that we're going to manipulate, us investigators. We're going to do things. We're going to set up an experimental group of chickens, and we're going to set up a control group of chickens. All right, so that goes on the x-axis because that's what we call the independent variable. The independent variable typically is the x-axis in a graph. I'm not going to write independent because I'm not going to be able to get that all on. But in our particular case, once again, it's two groups of chickens. So chickens, really. We got chickens over here that got injected. So these chickens were injected, and these chickens were not injected. So this is the not injected chickens. Okay? Independent variable is the variable that is standard, consistent, or it's the one you manipulate. Now, I don't have room on there, but I go ahead and say it loud enough. I think people will understand if I draw this one. For you guys just sitting here in class, if you think about weather, let's say month and temperature, okay? January through December. That goes on the x-axis. Right? That's the independent variable. Now, if you think about it logically, you're confused which one looking at a study is independent or dependent. So we're looking at temperature, air temperature. And everybody knows what it does here in Florida. January it's cooler, it's warmer in the summer, cools off again in the winter, right? All right, now let me ask you a question if you were going to look at that. Now, if you're listening to this online, what I've done is I've drawn a graph. Along the x-axis is the months, January through December. Along the y-axis is temperature, and I show you that it gets hotter in the summer. No earth-shaking science going on there, right? Let me ask you a question. Is the temperature of the air dependent upon the month, or is the month dependent upon the temperature of the air? One of those makes sense, and one of those makes no sense. Okay? Which one is it? Is temperature dependent upon the month, or is it the other way around? Yeah, exactly. It's hot in August because that's the month it is, right? It's not hot. It's not August because it's 87, right? You see what I mean? One makes more sense than the other one. So if you look at this, and again, typically in science, the x will be the independent, and the y-axis will be the dependent variable. The y-axis is the result of what you have along the x-axis. The temperature in January, the temperature in March, the temperature in August. Okay? It is supposed to say dependent. So if you look at it in terms of graphically, the dependent variable, right, the y-axis depends on what happens down here, right? It makes sense. For chickens, we'll take a look for a second. I've already given you the independent variable, right? I said you have a group of chickens that have been injected, the ones on the left and the ones on the right with the end, so they have not been injected. That's your control group on the right-hand side there. What is the dependent variable in this? There you go, whether they get sick or not. Absolutely. The dependent variable in this particular case is whether the chickens in each group or just one group get sick. Yeah. Because remember, if you phrase it sometimes in a sentence, it makes sense. Is the chicken, is the sickness dependent upon the chicken being injected or not? Or is the chicken being injected or not dependent upon the sickness? Again, one of those statements makes sense, the other one kind of makes no sense at all, right? That's how you tell dependent and independent variable. So, chickens being sick, and now, dependent variables, and remember, in the dependent variable, there's two of them, sick or not sick. Right? It's not just one variable. Again, what group of chickens in this particular example would be called the control group? Which group of chickens were the control group? The non-injected ones. They were the control. They were normal, everyday chickens that we didn't do anything to. We just set them right there and said, y'all do chicken stuff, and then you chickens over here, you do chicken stuff like they're doing, but guess what? We're going to give you a shot. Okay. Any scientific experiment, just in a short phrase, what is the purpose of a control? You can give me one word, actually, would sum it up, to be honest, if you think about what we're doing here. What's the purpose of a control? To make it equal except for that one thing you're testing, right, so that you can do what? There you go. The purpose of a control is to be able to compare things, right? You can't compare, you can't say, he's taller than what, right? There's no comparison there. The other day I told you about my friend doing the work on the dolphins, right? The dolphins hanging around in the Indian River, and the control was the ones that hung around in the restricted area by the Kennedy Space Center. That was the control. That wasn't a perfect control because there are humans down around there, but there's a lot fewer humans down around there. All right, now here's a good one. And this is the reason why you have to have a control. And we were doing a discussion in class the other day with pharmaceuticals, I think, right? Talking about testing pharmaceuticals. So I give you the pill that I think is going to help with your disease, you get better, or maybe it's a placebo effect. What do? I give the other person a sugar pill, right? But if they get better than placebo, if they don't get better, well, then it may be the drug, right? So take a look at, or think about that for a second. and answer this question. What mistaken conclusion, scientists don't always come up with the right answer the first time. Rarely do they come up with the right answer the first time. What mistaken conclusion could they have made had they not had the control group of chickens? In other words, all they had done is taken chickens into a room, injected them with blood, and then they got sick. What mistaken conclusion? Well, you don't have anything compared to, but that means you make a wrong decision. See, they did have a control group, so they said, wait a minute, the chickens that didn't get injected, they got sick too, so it's not a bacterium, right? But if it hadn't had the control group, remember, the injected chickens got sick. Okay, so if the injected chickens got sick and they didn't have a control group, they would have said what about their hypothesis, their original hypothesis about being a bacterium? I'm sorry. It would have supported the hypothesis, wouldn't it? Remember, support and refute we talked about? It would have supported the hypothesis that it is a bacterium that causes the disease. Now, you throw in the control group who weren't injected, right? Who weren't injected. They weren't exposed to the quote-unquote bacteria if it exists or not, and they got sick too. So what? We refute our hypothesis. It's obviously not that. And then at the very end, I asked sort of a question. Was the experiment a failure? Why do you say it wasn't?
It's kind of like a setback, even though it didn't support the hypothesis.
Right.
Now they know where they are, and they make changes. And that's the beginning.
You would know the hypothesis, right? Yeah, absolutely. I mean, down here, when you talk about an experiment being a failure, did this experiment fail to support my hypothesis? Yes. But the experiment is not to test whether it's trying why the hell do people get sick? That's what you're trying to figure out. Okay, so what you do is then you go back and you revise your hypothesis. So no experiment's really a failure. I always tell students, like, first time you got on a bike, did you ride it perfectly? I didn't. I ended up in the blackberry bushes. That's the first thing I did, right up the side of the road into the blackberry bushes. But I can ride a bike now. So I couldn't say, well, hell, I honestly can't ride a bike. That part of me is a failure, so I won't move on. No, I kept trying, and that's the same reason, because that's just the way science works, folks. That's the way science works. You test the hypothesis, you refute or support it, and then you revise it if it hasn't been supported. Hopefully, eventually, you come up with enough hypothesis that every time you test it comes up with pretty much the right answer. You predict it, and you can make that a theory. Make sense? All right, now that's only part of the online for your first assignment. That's the first part. I do have a second part, but if you paid attention and you understand that first part, the second part won't be any problem at all. What I'm going to do in the second part is they do another experiment. They revise their hypothesis, just like you said they should. They do another experiment, and I ask you similar questions about that as well. Okay? All right. Again, I'm sorry that it has to be done this way. Usually it's a PowerPoint, so it's no big deal, but today, you can even see it on the screen, it was not being cooperative. That's okay. I don't need you, computer. Okay, we talked in class the other day about the next set of slides, so I'm not going to show them. We talked about the fact that living things have a sense of organization, and I show you all the different wonderful ways that you can see that. We talked about, for example, in flowers that were organized. We talked about the need for metabolism. Well, let me show you this one, though, because I have a neat little graphic. So metabolism, and again, down at the bottom, remember the purpose of metabolism is to kind of keep us even on the seesaw. Back and forth, you get too hot, you sweat, you get too cold, you shiver, you don't eat enough, you eat, all those things that maintain balance. So I put this little phrase here to kind of help you remember the whole idea behind homeostasis. You got to remember, too, that entropy is the opposite of keeping things together. Entropy is when they fall apart. Everything falls apart, right? Your car falls apart. Your house falls apart. Everything does. Homeostasis, or metabolism, bringing in food. This is a hawk moth. They're huge. Sometimes people confuse them with hummingbirds, but this is the hawk moth. This is a polar bear, right? Obviously, polar bears have to eat. Interesting enough about this is just kind of a side. Polar bears obviously have lots of adaptations to stay warm, but one of the ones I found most fascinating is recently they did research where they sliced through a polar bear hair. So this little picture right here. Everybody online is going, why is he pointing to? I'm talking about that right there. That is the cross-section through the hair of a polar bear. You notice it's hollow inside, provides insulation. So that air inside is warmed up by his body, provides insulation to the outside cold. You don't have holes in your hair, but you don't need it, right? Anyway, these organisms, wherever they live and how they do it, are trying to maintain that balance, trying to maintain homeostasis mitigates entropy. In other words, you never defeat entropy, it mitigates it, slows it down, keeps it back for a while. Eventually, it always wins because that's just the way it is. Nothing can stay organized forever. I mentioned the other day in class about evolution. Now, I don't talk a lot about it in here because actually I spent an entire, about a month in Bio 2 talking about it, but I do want to give you the basic parameters of it so at least you all understand. This is probably one of the most misunderstood theories, and that's some of the most misinformation of scientific information out there, and it has a lot of implications socially, and I imagine that's why it's so hotly debated. Anyway, this is the way it basically Charles Darwin was telling us this is the mechanism behind evolution. See, organisms have changed through time. We know that. The dinosaurs aren't here. We can look in the fossil record and see there are creatures that used to be here aren't here anymore. Right? We can find stuff that there are no Tyrannosauruses wandering around. They went somewhere. So if we take a look at these organisms and look at the mechanism by which all that happens, we take a look at first a population of organisms. Now, a population has a specific title or term or definition. A population is groups of conspecifics, yes, I use my mouse with my right hand, but I'm left-handed, so this should come out really well. Specifics. You can spell the rest of that. Conspecifics, members of the same species, okay? Members of the same species. Who have the potential to breed with one another. That's what a population is. If we kind of go outside the human realm for just a minute to think about some of the other critters, the squirrels that hang around campus up here on Lake Sumter and the squirrels that hang around Lake Eola in downtown Orlando are both all the same species. They're conspecifics. They're eastern gray squirrels. Okay? But they're not the same population. And the reason why they're not the same population is it's not reasonable that they could breed with one another. See, the ones up here are going to breed with the squirrels hanging around campus. Well, nobody's going to go from downtown Orlando to Leesburg, to breed with a squirrel. There's plenty of squirrels downtown. So it's all about the genetics. So the genetics up here stays Lake Sumter squirrel genetics. And that might be a little different. Not a whole lot different. They're still squirrels, same species. But now we got Lake Eola of genetics. I've traveled up to Maine over the summer, and I'm always amazed when I go up north. And those of you who have ever been up north or lived up north know that the squirrels up there are a lot bigger than the squirrels down here. Right? They're huge. They're like dogs sometimes. I was up in Maine, and I was walking through a forest trail. I was like, what the heck? It was a squirrel. It was the biggest squirrel I'd ever seen. Now, again, there are reasons why they're bigger up north. It has to do with them staying warmer, but nonetheless, those are separate populations. But they're all eastern gray squirrels. They're all the same species, okay? So a population of organisms, same species, and they can breed together. Now, there are two things about organisms that we have. Hereditary variation, we're all different, right? Remember the other day we did sexual reproduction, A, B, and maybe asexual, different story. Asexual organisms do evolve, but they have a different way they go about doing it. So we have hereditary variation. What this is, we have differences. Back during the Dark Ages, 14th century, 13th century, a flu spread throughout Europe, the world, right? The bubonic plague. You've all heard about it, right? How many people in Europe did that kill? What portion of Europe did it kill? You know? I don't know because I'm an expert on disease. I know because I just saw a show with someone who said 30% of the European population died from bubonic plague. 30%. That means if we're in this room, one of us is dead. One of us is dead out of this room. So if you think about your family, your community, count 1 in 3 people is going to be dead. Right? That's amazing. What is incredible? There were reports that investigators would go around to villages, and the entire village was dead. Everybody, all 200 of them were dead. Sometimes they go to villages and all of them are dead except for a couple of them. They go to another village and here's another guy. Now, you were exposed to the plague. There's no way you lived in one of those villages in which 200 people died, and you over here who happens to just still be alive and healthy and doing fine, you had to have been exposed to the plague. How come they didn't die? How come they didn't die? Did you get measles when you were a kid? You didn't? You know, they get shots for that nowadays, don't they? I keep forgetting that measles are childhood diseases that we all got. We all got them. It was like chicken pox back then. You get the measles when you were a kid? You don't know? Yeah, well, when I was a kid, there was this really barbaric thing that mothers used to do, especially when you were preschool or right before you started getting into first grade. Those childhood diseases like measles and chicken pox, you would want that child to have that disease because you develop immunity before they went to school. Mothers may not have known back then, but there also is the idea that having them younger is better for you. If you had chicken pox and measles as an adult, measles can actually cause heart disease, heart failure in some older folks. So you get sick, you get immune to it and all that sort of stuff. So when that was spreading around my neighborhood, that's what my mother did. She brought me next door. There were a family. I remember 5 boys in this family. And the boy that was about my age, the guy I played with, was a guy named Michael Hawes. Now this is 50 years ago. I wonder if this person's ears ring every time. Why I would remember his name, I don't know. Michael Hawes had 4 brothers. They all got the chicken pox. I got the chicken pox with them. Michael never got the chicken pox. Never got the chicken pox. Now, he was exposed to it. He wasn't immunized against it, but he was exposed to it. Come on, he lived with 4 other snotty brothers. You know that he had been exposed to that virus that causes chicken pox. Why didn't he get it? He's lucky, right? He just happened to be born with that genetic variability that he was naturally immune to that. And that's what happened with the plague as well. So when we look around to a group of individuals in this hereditary variation, we realize that in some cases it's not just about, oh, he's a little taller, she's a little shorter, his hair is darker. It's not just about those aesthetic kind of things. It's about life and death sometimes. So when diseases go through a population, there might be some folks that are naturally immune. I do a thing in my Bio-2 class where we take a petri dish and try to grow bacteria. A common thing. Kind of show everybody that there's stuff going everywhere. People don't usually get used to that. So they do chairs. Cell phones are popular places for them to do. The counter up here, of course. And you can imagine, bathroom, toilets, remarkably clean. Amazing. All right, I guess our cleaning staff does a hell of a job because every time I go down to the bathroom, you would think, right? Remarkably clean. One of the dirtiest places, and those of you online, I'm over here by the purification thing. Little thing you squirt with your hand, you put purification. That's one of the dirtiest places around with the bacteria. Because guess what we're doing? We're killing off lots of the bacteria and stuff. But we ain't killing off all of them, are we? There's a few of them that are hanging around. We're building superbugs and bacteria. In my opinion, those things might actually be worse. And mothers who douse their kids and that stuff when they go out, I get it. I understand you're trying to protect your child. You might not actually be protected. You need to be exposed to that stuff. To develop immunity to it, okay? Anyway, so we're all different. Some of us might die from the plague. Some of us might not die from something, but maybe we don't live as long. Okay, for whatever reason. We have sickle cell anemia. We have some disease that may shorten our life. Regardless of all of that, if you don't live as long as somebody else, you can't make as many offspring. And people can't reproduce. What's that? So if we're different, and if you look over on the right-hand side, one of the things that Darwin recognized when he was traveling all over the world was, oh my goodness, we can make a lot more offspring than can possibly hope to survive. Not a big thing in humans, at 8 billion, but if you think about some other creatures like sea turtles. Oh, there it is. Sea turtles crawl up on the beach. Each female lays about 100 eggs. They're doing it this time of the year. I was at Sarasota this summer, and there were nests. You've been on the beach this time of year. You've seen them too. They have them marked off with the sticks and all that sort of stuff. But sea turtles crawl up on the beach, and a female lays about 100 eggs in a clutch. That's what we call a group of reptile or bird eggs. It's a clutch. They dig a big hole, and they drop these about ping pong size eggs in there, right? 100 of those. Now, those about 50 days later will hatch. Now, if that's something you've never seen, and you can get with tours, they'll take you out and do that, you've got to watch sea turtle hatchlings happen. It's amazing. They're on the beach, and they're everywhere. So little tiny hatchlings crawling, and of course they follow light. That's the problem with light. So you got your little flashlight, and you kind of, oh my God, you're like Pied Piper of sea turtles, man. You're leading sea turtles everywhere. Lead them to the water, right? Anyway, of those little hatchlings, out of that 100 that mother laid, the chances are none of them will live long enough to reproduce themselves. None of them. Actually, the odds are it's 1 in 1,000. You need 10 sea turtle nests for one egg out of those 10, which was about 100, like I said, to live long enough to reproduce itself. So genetically, its information is gone, is what I'm trying to say. Right? So when we think about that, why do sea turtles have so many offspring then? You only know 1 in 1,000 is going to make it. You better make sure you spit out at least 1,000, right? So what we do is we overproduce offspring. I worked on an eel when I was in college, and the females during breeding season could produce up to 250,000 eggs. 250,000, they breed twice a year. So they would produce 500 million eggs in a year. Males can produce tons of sperm, and there were plenty of eels. So tons of eels making all these offspring. So what Darwin says is, we're making more offspring that can possibly help us survive, so guess what? We're going to compete. Because not all those offspring are the same. Some are a little faster swimmers if we're thinking about the sea turtles. Now, if I'm a little faster swimmer than you are, guess who's shark food? Guess who's shark food, right? The guy who can't swim as fast. Just like we said the other day, out wandering around the woods, you come across a wild animal intent on eating one of you or one of you in the group. What do you got to do? You just got to not run somebody in the group, right? You don't have to run everybody. When I take my son scuba diving the first time, he said, why do you carry a dive knife, Dad? I said, what do you mean what I carry? He said, why do scuba divers carry a dive knife? And I said, if there's a shark, I'm going to stab you. That's the whole purpose of it. Anyway, so you get the idea, right, that there's a lot of offspring born. They're all different, and it's not just they're pretty or not pretty, but they're competitive or they're not competitive. Now, being competitive means you live longer, you get more food, you have more offspring. Okay? And if you have more offspring, that means that those of you that are successful are going to leave behind more offspring. All right? So you live 20 years and have 3 children. Okay? You live 40 years and have 10 children. It's a numbers game. So as we go into the future, what happens is the genetics of the successful one is the one that passes down through into the future. This is what we mean by a population adapting. Be very careful. Evolution just indicates that life on Earth has changed. Right? We knew all about that even before Darwin. Darwin gave us this mechanism by which it changed, this competition, this idea that those get with reproductive success because of the fact that they have some variation and they can compete. Those are the ones that leave behind the genetic. If you want to know more about that, like I said, I spent about a month on that in Bio 2. Well, that and other aspects of that. But that's it's a theory with a whole lot of really interesting implications. We answer a lot of questions about why life is on Earth the way it is. Oh, we got the giraffe response. Let's go down. Everybody knows what this is, right? Everything on Earth that's alive has this, right? Everything on Earth. Everything on Earth. We're all unified by that. Biologists always love to talk about this unity and diversity of life. There's how many different species? Not counting the ones that aren't around anymore, but all of them have DNA. To this point, we have not found a life form that can survive without DNA. There was some thought that they found some bacterium living in some really weird hot spring somewhere that appeared not to use DNA, but later research showed out that that wasn't the case. To me, from an astrobiology point of view, life on other planets, possible? What do you think? It's certainly possible. I mean, they tell us that there's as many stars. There's as many stars in the universe as there are grains of sand on all the beaches on Earth. Lots of stars, lots of galaxies, right? So probability-wise, we would say that. From my point of view, and my interest in astrobiology, I love when they do that, is a DNA life. If we find life on Titan, or one of the moons of Saturn, one of the moons of Jupiter, or even on another planetary body, does it use DNA? It'd be fascinating to find out, because as far as we know, considering all the species on Earth, there's billions and whatever have been and are, it's always been DNA. It'd be fascinating to see if somebody else, some other creature has figured out a different way to do that. That's what's fascinating about astrobiology. I mean, it was, oh, we're going to make Mars. No, it's probably single-celled organisms. right? If we find life in this solar system, but that's still pretty fascinating, if you think about it. To this point, we only knew there was life here. We may not be alone. All right, let's look at some of the organization aspects of organization of life on Earth. I want to start backwards. Sorry, I'm a person that does that the other way. Get that out of the way. Stop. So I'm going to start down with number 10. So we're going to go small to big. I like going small to big. So what's interesting about this to me is not only the biological part of it, but we bridge the gap between the non-living and the living, between the chemistry and the biology, right about here, right? Molecules can be all kinds of things. Water is a molecule, it's not alive. Carbon dioxide is a molecule, it's not alive. Carbohydrate is a molecule, it's not alive. Right? Molecules are not alive. But when special molecules and special arrangements come together to make up parts of a cell, we call organelles. Now, it's an interesting word because it sounds kind of like organ. We have organs, cells have organelles. Right? Now, see, and those organelles can be things like that you're familiar with, mitochondria, nucleus, ribosomes, endoplasmic reticulum. Remember those terms? Oh, that's cool. I love that kind of stuff. Anyway, those organelles make up a. Now, the other day we were talking about all the things that make something alive. Metabolism, response, reproduction. It is not until you get to the cell level, until you work your way up to this level here, the cell level, that you have a living thing. Organelles are not alive. All right? Molecules we talked about are not alive. It's not until you get to the cell is the smallest entity that has all those characteristics of life. It's organized, it metabolizes, it evolves, it responds. You poke a cell with a needle, and we'll move away from it, and it reproduces. Self-divides. Right? So that's, prior to that point is non-life. Now, cells make up tissues. If you go into A&P, you'll learn all about tissues. There are various different types of tissues. Tissues are, what's the word I'm looking for? Tissues are the thing about animals. All right. Now, tissues occur in plants and other things, obviously. But among animals, tissues became very specialized. Tissues became very complicated in the structure. We have nervous tissue. Plants don't have nervous tissue. The plants don't hurt and cut them, right? They don't feel anything when you talk next to them. If you talk next to your plants, I like this. See how well it grows? I said, you could take it through simple other things. It will grow the same. It's the carbon dioxide that you're putting on the plant, but it makes her feel better, and that's fine. But they don't have nervous tissue. They can't think, right? So while we find tissues, again, it's kind of a bio-2 thing as well. It's only when we get to animal tissues. All right? Tissues make up organs, and then organs make up organ systems. So you have the stomach, the intestines, and that's all part of it. Then those that will make up, obviously, organisms. And then within organisms, we have communities of organisms. What are communities? Organisms that interact with one another. Right? So we have a forest, we have the trees, we have the squirrels, we have the worms, the birds, the bacterium, the fungus, all those other things, the protozoans, let's not forget those, are often interacting with one another. Right? So a predator eats a prey, that's an interaction. Not particularly pleasant for the prey, but nonetheless. Clownfish live in the tentacles of the sea anemone. That's an interaction. Right? So they're interacting with their community. Now, when we talk about communities, the interactions are going to occur only among living things. Only among things that we call collectively biotic. B-I-O-T-I-C. Biotic. So in a community, we have all the biotic parts. Do living things interact with things that aren't living too? I mean, not just predator, prey, all that stuff we talked about, but do they interact with non-living parts of their environment? Yeah, but think about it in terms of like carrying on life processes. Is there something you're doing with those basic life processes, or you're engaging with the parts of life that aren't alive? Inhale. Inhale. What is that? You just inhale. Mixture stuff, right? Any of that stuff alive? No, gases. There might be a few bacteria, but it's all gases, right? Oxygen, not alive, you interact with it. And then you give off carbon dioxide. And guess what the plants do with your carbon dioxide? Photosynthesis, right? So they're all interacting. Or one that might be a little more obvious, do fish interact with water? Yeah, and that's not alive, right? Water's not alive. So sure enough, living things that are biotic also interact with, and I'm putting an A in front of biotic, with things that are abiotic. In science, often putting an A in front of a word means the opposite of that. A or an AN, depending if the word starts with a vowel. So now when we get to the point of the ecosystem, then we're talking about all the living things and all the non-living things and how they interact. Rain, temperature, gas exchange, nutrients in the ground, nitrates, phosphates, things that we call fertilizers. When you fertilize your lawn, you're doing an abiotic biotic thing. The lawn is alive, and you're putting chemicals on it. They're not alive, but they interact because your yard gets better, it gets greener, it grows. And then when we move all the way up onto Earth, we have the biosphere. I like that word. I think that's cool. Life sphere. Now, when I first started teaching this class, I used to say, they tried to build a biosphere here on Earth. They tried to actually build a unit of Earth. Think about it for space travel. They were trying to do research to see if we could get to other planets, if you could build Earth on another planet. It should be easy enough to build Earth. I mean, after all, we kind of know all the parts, right? It's like I tell my students, building Earth, or even scientists, would be like if we came to the lab next week and decided we were going to build a nuclear power plant instead of the lab. How many of y'all know how to build a I'm sure I'm like, no. We probably wouldn't make a very good nuclear power plant, right? We wouldn't be very good at that. So they built this other, they called it Biosphere 2, and everybody goes, oh yeah, the Pauly movie, what's his name, the comedian was around years ago, he made a movie about it. And I'm like, no, not the movie. This is what I know I'm getting over before. But oh yes, it was out in Arizona. We remember reading about it. They put 6 scientists into it for 18 months. That was the idea. We'll lock you inside for 18 months. No exchange from the outside world. No food was going to come in. No waste was going to go out. No gases were going to come in. No oxygen. No carbon dioxide. The plants were going to take care of that. It was going to be a terrarium, right? Big, huge terrarium. So they locked these scientists in there for a total of 18 months. They lasted about 6 weeks. When they didn't die, they took them out because the carbon dioxide levels began to rise so quickly inside it became dangerous for them to stay there. You must have missed something about the way the Earth works, right? So building an artificial Earth, Biosphere 2, nice try, but it just shows how much we don't know about what life is doing. In spite of the fact we seem to know a lot about it, we don't know enough about to create this living biosphere. Someday maybe, but certainly not right now. There's a lot more to know. Would you have done that? Let yourself be locked away in that for 18 months? Just you and 5 other people, This doesn't with my family. But if there's like people I like and stuff that long, getting away would be the hard part. Getting kind of yourself, crawling off into the cornfield. Like they did. If you looked at it, it was cool. You can still find pictures of trailers pointed down. They had a little cornfield, they had a little coral reef. I mean, they had everything. They just thought it was a little more difficult than they had anticipated, which I'm glad, because it means that us biologists still have a job to do because we haven't figured it all out yet. All right. Here we go. Not going to spend a lot of time on this. Really isn't much to it, right? It's basically taxonomy. I know that some of you have probably seen this before. Taxonomy is the science of classifying organisms, right? This is the whole kingdom, phylum, class, order, family, genus, and species thing. You may not be familiar that we do have now something bigger than a kingdom. It's called a domain. We'll talk about what the domains here are in just a second. But it is hierarchical in the sense that phylum fit into kingdoms and classes fit into phylum and orders fit into classes and so on and so forth. So we might have the kingdom Animalia, which includes all the animals. A lot of animals out there. We have a phylum This is not a zoo class, not a biology class. I don't expect you to know the names of these groups. I'm just showing those examples. But among the kingdom Animalia, we have the phylum Chordata. We belong to that one. Phylum Chordata, individuals with a backbone, vertebrates. Vertebrates are the class. Not all animals are vertebrates. And then the class Mammalia, we talked the other day a little bit about what it means to be a mammal. The family Ursidae, which is kind of cool. Bears are one of my favorite critters out there. And then there are different types of bears. And then down at the very end, there is one name for the one type of bear, right? This is Ursus americanus. Now, there are many other Ursids out there, but this one bear, the black bear, Ursus americanus, right? That's its binomial two parts, right? Ursus, the genus, and the species nomenclature. Now, these things, when they're put together, originally the scientists that put this all together just kind of did it by, well, the way they look. And he actually, on record, is saying that this doesn't mean anything about the way they're related. Just because you're in the same kingdom or class or phylum or whatever, doesn't mean you're related to everybody else in that group. We know now that's not the case. We know now that there are genetic relationships between these critters. And so if you look at it, members of a genus would have a lot more similarity genetically than the members of the class, right? The members of the genus Ursus would be all bears. In the class Mammalia, holy cow, you got the dolphins and all these other things out there. So now we know that this does, or we have with our data suggests us, or shows us that that is something to do with relationship. It may, the domain may be the one that you're not familiar with. So let me, there we go. That's a new one that they added to, because we found out something about little tiny critters that we just used to lump them together and call them little tiny critters. That's not a good way to identify things, right? Think about protozoans. Protozoans are all microscopic and drop of pond water and all that sort of stuff, but we all lump together and call them protozoans. Now, the kingdom protozoan is defunct. We're still using it, but there are suggestions there may be up to 20 kingdoms of protozoans. So think about what that says. There are differences between two protozoans as much as between you and kingdom Animalia and plants in kingdom Plants. So if you're into new stuff, protozoology, man, that's the thing to go. So there's the kingdom Protista on the lower right. I put quotes around it because I want you to understand again that is not really an official thing. Kingdom Animalia, and of course the idea again is you share characteristics. Animals have active movement, where we don't photosynthesize, very simply. The plants, of course, photosynthesize and don't have active movement. What do these guys do? How do they make their living? How do mushrooms make their living? They don't photosynthesize. It's got to be green to photosynthesize. I thought that'd be cool thing. Right? Just think about it, you driving down the street, you always have time to stop and something. I'm not doing this. What are the funny guys do? Decomposers. They're decomposers. They break things down. Actually, fungi are weird in the situation that we bring our food on the inside and we digest it, right? They actually, we have enzymes that do that. They actually, you can't spit up, don't have a mouth, but they release those enzymes into the environment, digest the food, and then suck it up. A little bit different than the way we do it. This is a puffball. I found it on campus. This is a mushroom. I am going to do, let me turn the video on for just a second. There we go. This is a puffball mushroom. I don't know how well you can see. You guys will be able to see. Feel that? It's like a gas, doesn't it? Those are spores. Spores. Millions and millions of spores. That's the reproductive unit of this thing. I was talking about 100 eggs. How many do you think I'm squeezing out at any one time here? Tens of thousands, right? These things can make millions and millions of spores. So fungi, weird organisms. All right, now the ones that you may not or you learned collectively, at least I did when I was in school, as another kingdom. We used to call it Kingdom Monera. What was it? 8 to 9, 20, right? We used to call it Kingdom Monera. Don't write that word down. No such thing as Kingdom Monera anymore. We used to put these guys, the prokaryotes. We'll learn about what prokaryotes are in a couple of chapters. But we used to lump those together in the Kingdom Monera. See, everybody else down here, the domain Eukarya, all right, that's these
4 kingdoms. The domain eukarya are considered eukaryotes. I know I'm getting a little ahead of myself, not too ahead, because in just a few chapters we will introduce those 2 terms. But what I'm trying to point out is we put all the prokaryotes together in one kingdom because they were small, because bacteria are small. Now we find out that there is so much difference between these prokaryotes, and now we've broken them up into 2 domains. So we have the domain bacteria, and then we have, my favorite ones, the extremophile prokaryotes. These are the archaea, which means old and ancient. We think that these are perhaps some of the closest descendants of the first critters on the planet. They can live in boiling water above 100 degrees Celsius. They can live in acid of 0.02 pH. Not, I don't know how much you guys know about the pH scale, right? But you get away from 7 towards 0, it becomes very more and more acidic. Something that has a pH of 0, really acidic. So they can live in that. A 0.2 acidic solution would dissolve metal, and they live there, right? So some interesting, really, ideas about how these critters have these incredible adaptations to live in these extreme environments. And astrobiologists, again, could they travel on asteroids? Because they can survive radiation well beyond what we can do. So that's a really neat group of organisms. All right. Phylogenetic tree of life. Just to kind of show you, let me close that thing on the right. Just to kind of show you where we are. I always like it when you go to the mall, you know, you are here. So this is our universal common ancestor, UCA, whatever that was, some single cell critter that was back, you know, when life began to take hold on Earth. That would have been the first time, like I said. So then we break off, and here's the domain bacteria now. These are all the different types of bacteria. They have all the kingdoms and phylums and all that stuff. My favorite, the archaeans, Methanococcus, they can live off methane. So when they say things like, oh, there's lakes of methane on Titan around Saturn, that could these organisms could live in methane here on Earth. So they might be able to live out there on methane in those as well. Thermoproteus, whenever you see thermo, these are the ones that can live in extreme heat, like boiling water conditions. The halophiles, halo salt, foul to love, they live in really salty conditions. So that's what I mean about eukarya being kind of the X games of prokaryotes that can survive. And then all of us are over here, right? These are all the eukaryotes. These are some protozoans that are stuck in there because, again, it's a weird group. Animals and fungi, you are here. Right? Notice, though, interestingly enough, among the prokaryotes, this indicates to us that you and I are more closely related to those really primitive, not a great word, ancestral type of prokaryotes than we are even to the more descendant type of prokaryotes. We're actually more related to the extreme part than we are to the other ones. I think that's it for that. Yeah. All right, folks, we'll call it a day for that. We're a little bit early, but we're on schedule, which is good. I wanted to get through chapter 1 this week, and that's what we did. Start working on that scientific method assignment. Like I said, we've already talked about it here. If you have any questions or problems, get in touch with me. And again, for everybody listening otherwise, make sure you check the book downloaded, check Blackboard, make sure all those problems are worked out, okay? Any questions for me? Anybody got? Oh, all right. See y'all later. Thank you for showing up.